Now, if we want to perform gene manipulation, then the first step, of course, it is the isolation of genomic DNA. और ये जो genomic DNA है, that represents whole gene complement of the organism. It also include our gene of interest. तो सवाल ये है कि हम कैसे DNA isolate कर सकते हैं. So the basic step is that, for example, if we want to proceed with E. coli, that is the microbes, then the microbes like E. coli, they will be grown in a suitable culture medium, and then they will be incubated for 24 hours. After 24 hours, visible mass of E. coli cells or any microbial cells will be produced, and then the cells will be separated or harvested by centrifugation. जब सेल पैलेट या सेल बायोमास सेपरेट हो जाए तो फिर इसको फर्दर प्रोसीड किया जाता है फॉर द आइसोलेशन ऑफ genomic DNA. Now, different type of chemical reagents are available by which we can lyse the cell. सेल को हम डिसइंटीग्रेट कर सकते हैं, तोड़ फोड़ सकते हैं, like हम STS यूज़ कर सकते हैं, that can causes the lysis of the cell, and when cell is lysed, then different type of proteins, they can be disintegrated by using proteinase K, and after that chloroform or phenol can be used for the extraction of the genomic DNA. जब genomic DNA extract हो जाए तो finally इसको precipitate कर लिया जाता है by using another solvent which is isopropanol. So this is the manual method. Nowadays different type of commercial kits are available from different suppliers, companies, and by using these commercial kits we can isolate genomic DNA within 1.5 hours from any cell type. चाहे ये बैक्टीरियल सेल है या प्लांट सेल है, एनिमल सेल है या blood cells हैं. So nowadays it is very easy to isolate the genomic DNA, and once it is isolated, then it can be dissolved into a suitable buffer for its maintenance for further experimentation. Now, once genomic DNA it is isolated, then how it can be analyzed? कि DNA isolate हो गया है, हमें कैसे पता चलेगा कि हमारे एक्सट्रैक्ट के अंदर DNA जो है वो isolate हो चुका है?